today is Christina Harris-Jackson. Dr. Jackson is the Chief Operating Officer and part owner of Omega Quant, and she is an assistant professor in the Department of Internal Medicine at the University of South Dakota Sanford School of Medicine. Christina received her PhD in nutritional sciences from Penn State in 2013 and completed her training to become an RD in 2014. Christina continues her research in the area of omega-3s in maternal health, helping create the prenatal DHA test and the mother's milk DHA test. And she is going to be talking today about how testing hemoglobin A1c, vitamin D, and B12 levels can support a healthy brain. So I'll go ahead and turn it over to Christina. It's all you.
Thank you, Kelly. So as she said, just a minute, stop sharing. I need to reload this. She said, I am primarily an omega-3 researcher. So it's always fun when I have to not talk about omega-3s and challenging. It takes me back to my doctorate days. So this was interesting to get back into these different nutrients and processes. So basic objectives are talking about the vitamin D, B12, and basically blood sugar, HbA1c, and how they relate to brain health, and how nutrient status is important for maintaining that brain health. But brain health is a super vague term. So once I started to get into the research, I found that there was a lot around the aging brain, and primarily focused on Alzheimer's disease and dementia. So for today, we are going to talk about those two things. And I should note that Alzheimer's and dementia increase in risk with age, but it is not necessarily normal aging. So they are both disease states. From the Alzheimer's organization in the US, we have this helps kind of put them where they need to be. So dementia is the larger term that encompasses conditions where people have a decline in memory, reasoning, or other thinking skills. Alzheimer's is more specific brain disease, and it counts for 60 to 80% of dementia cases. So the dementia is the larger aspect, but Alzheimer's is really climbing. I am not going to go into the details of the mechanisms of Alzheimer's and dementia. That is far too much, but we will just showing you this picture of these two brains. One is a healthy aging brain and one is an Alzheimer's brain. And we can see there, the Alzheimer's brain is shrinking. So we'll look at some research that looks at brain atrophy or brain shrinking. That's one of the physical signs of it. You can see there are plaques, diseased neurons, tangles. It's pretty clear that this brain is kind of starved of the nutrients it needs in a variety of ways. And so that's part of when we're talking about holistic health. It's you need all of your systems functioning well for your brain to stay healthy, your cardiovascular disease system, inflammation, having enough nutrients on board. So this is the general picture we're going to think about as we go through today, instead of getting into the weeds on Alzheimer's and dementia's mechanisms. And just like almost everything else, risk factors for Alzheimer's and dementia are very similar to other chronic diseases. In fact, having other chronic diseases increase risk for having this brain chronic disease. So having, we have modifiable risk factors, things that we have some level of control over, maybe not 100% control, but some level. So diabetes, obesity, cardiovascular disease, traumatic brain injury, pollution, lifestyle, and smoking. All of these have links to increased risk for Alzheimer's. And then things that we can't change: age, sex, race, and genetic mutations and polymorphisms. There's definitely large, and there's also a lot that we don't yet know, whether it's modifiable or non-modifiable. So it's important to keep in mind what we can control and what we can't. Like all the other chronic diseases, the preventative measures for Alzheimer's and dementia are keeping your entire body body, mind, and spirit healthy as long as you can. Making sure your cardiovascular status is good, making sure you're getting blood flow throughout the body into the brain, eating healthfully. So that's where we're going to focus today when we talk about nutrient status. We'll talk a little bit about the dietary intake, but also want to talk about blood status because those aren't always the same thing. Making sure your life, you do things you enjoy in your life, keeping your brain active, avoiding knocks to the head that can cause that traumatic brain injury, social connections, hearing, and exercise. Hugely important is exercise, keeping the blood flow going. So all of these are pretty well known at this point, I would say, as far as what people know about preventing this disease and many other chronic aging diseases. So today we are clearly taking a very small slice of this whole picture, looking at vitamin B12 and as well as our MMA and creatinine tests, vitamin D, and the blood sugar control with HbA1c. We'll focus on those. And I just feel, especially when we talk about Alzheimer's and dementia, there is a lot of fear around this disease. It is growing. It can be very devastating to the person and their family. And then that kind of puts pressure on all this research. And I have to note that even if you do everything in your power correctly, you could still get Alzheimer's and dementia. Even if you don't do anything, you may not get it. Everyone knows the smoker that just did whatever they wanted and lived to 110 and was like sharp as a tack. So we don't have complete control over the disease state, but doesn't mean we have zero control. So when we're talking about preventative measures, we always talk about reduced risk. And in that, sometimes I feel people equate reduced risk with no risk. So if you're in the right category for all of these, it does not guarantee that you won't get any of these things. So in general, with patients, this is probably kind of hard beyond what you discuss with them, but it is really that serenity prayer of focusing on being empowered to live a healthy lifestyle where you can, and also understand where you don't have control. So it's definitely a balance. And when we are predicting these long-term chronic events, there are so many variables happening. We're not always sure which variable is actually causing the change. So that's just my little soapbox before we get into talking about these nutrients and the research around them. Let's start with B12. So real quick refresher. Also, B12 is also called cobalamin, produced by bacteria and found in soil and contaminated waters. But for humans, we typically get it out of animal products. Some plants are very high in B12. We also fortify our foods with it, and we can take a supplement. It requires intrinsic factor from the stomach to be absorbed, and therefore our deficiency state is either based on dietary restrictions or poor absorption or something else affecting B12 status in the body, like some medications or disease states. So here we can see we're talking about aging brains, malabsorption, and the whole GI system as it ages, just like our skin ages, that ages as well. So when we don't have, we might have issues absorbing B12 because of both the status of our guts as well as that intrinsic factor. So here in this diagram, you can kind of see where the B12 picks up the intrinsic factor, and then it needs that to be able to be taken up into the gut and put into the bloodstream. Transcobalamin is what transports it in the bloodstream. If you have more B12 than transcobalamin, you will pee it out. So this is kind of the B complex vitamins, the bright yellow pea. If you don't have enough transporters, you could have expensive urine, but that's okay. If there's inadequate B12 in the diet, the liver will start recycling it. Actually, you can recycle it for, I think, 10 years. So this is a very slow moving type of deficiency. And when that occurs, you may see a low to normal range result on the serum cobalamin test. And when you're in that state, that's when neurological symptoms can occur. The classic signs of B12 deficiency are pernicious and megaloblastic anemia. Before that happens, we start to see the neurological symptoms. So this is why testing can be really important. So why is B12 important neurologically? And for so many other things, it's a part of the single carbon metabolism system. And you guys have heard about all this. This is where folate comes in. This is where B12 comes in. This is how our cells repair DNA and synthesize DNA. So DNA is the source of all the proteins in our cell. If that is not, if the DNA is not healthy, the cell will not be healthy for very long. So this is a foundational system. You can see this part is the folic acid part of the cycle. B12 shows up here in the cycle. And so the main things it's doing are it's taking homocysteine and converting it to methionine. And then this cycle is able to occur here. The reason why homocysteine, which you've probably all heard of, is a signal of poor vitamin B status and also the poor health of the system is because when you don't have B12, this process doesn't happen. This builds up in your blood. So it's a sign that this is not working. On the other side, this is in the cytosol of the cell. In the mitochondria, B12 is also a part of a system involved in fatty acid processing. If B12 is not functional or available for this process, converting MM-CoA to methylmalonic acid will build up. So methylmalonic acid and homocysteine are both insufficiency markers for B vitamin status. If both of those are becoming elevated in the blood, your B vitamin status is insufficient or not functional. The MMA is more specific to B12. Homocysteine can also be affected by folic acid levels. So these are kind of the foundational systems why B12 is important. Also on this fatty acid side, this is also where potentially like myelination of nerves can come into play. A group of international scientists that are experts in the B vitamins and B12 in particular wrote a paper about homocysteine and dementia. And these are the people who really focus on that subclinical cobalamin deficiency that I'll talk about in a minute. This is really the neurological side of B12 deficiencies. For them, they have both folate and B12. Low levels of that increase homocysteine, and that leads to or is a signal of cognitive impairment. And the combination of all of these things are where they say this is really showing that the whole system can lead to cognitive impairment, and even worse, when you have this feedback loop. When you're cognitively impaired, you might not make the best lifestyle decisions. You might have a disease or have to take a drug that further impairs the system or impairs your ability to absorb or choose the foods that have these. So basically, they're just alerting that this homocysteine and the B vitamins are a major factor in brain health, and that needs to be noted. So for them, in their view of the research, they believe plasma total homocysteine is a modifiable risk factor for the development of cognitive decline, dementia, AD in older persons. So the logic would say if homocysteine is a risk factor, then the B vitamins, including B12, would play a role in the prevention. So here's a little bit deeper into the subclinical cobalamin deficiency. Over here, these are studies that have looked at subclinical levels and found cognitive deficit, Alzheimer's disease, and brain atrophy and depression all have shown that they are related to B12 status when it's in low to normal ranges. It's primarily affecting the nervous system at those levels. And this over here is kind of their argument against just looking at the classical deficiency signs of anemia, the metabolic insufficiency. So that essentially means your B12 or your folate, you either don't have enough or you don't have enough that's functional. And where does that start to happen? Here's plasma MMA and homocysteine. When they're high, that means B vitamin functionality is low, and that's where your true deficiency is very obvious when those are very high. But what happens when it's just starting to take off? And this is the area where they're focused on. This is where the neurological issues start to happen, and it's very hard to notice. And the same type of thing with the homocysteine. And so this is where their argument is to target the B vitamins at a lower MMA or homocysteine level. Individuals with B12 deficiency can suffer permanent neurological damage, even when the rest of their hematocrit, MCV, and even their serum cobalamin level is good. And non-anemic seniors identified with the mild B12 deficiency were determined to be at the 2.6-fold increase of risk for suffering all of these conditions. The damage is insidious, often occurring before noticeable symptoms. This is the key with B12, is how slow the process can be. So catching it early with testing is the best option. So what are our cutoffs for UMA for B12 deficiency and the subclinical deficiencies? So normal serum cobalamin for low to normal B12 status is 200 to 350 picogram per mil. A low level, like a deficiency state, would be less than 200. And also these numbers are also debatable, but this is typically what's used. If we look at the methylmalonic acid creatinine ratio, creatinine is because it's in urine, to make sure that it's normalized to that. That's a high ratio. That means B12 status is low. So multiple labs here, because this is not a super common test. I just wanted to put up all the different cutoffs different labs have. The lab that we worked with to develop this test is a Norman lab. He's a researcher in the area. They use a 3.6 millimole per mole creatinine as the cutoff. If you're above that, that means B12 status is impaired. LabCorp uses 2.8, Quest uses 2.2, and the Mayo Clinic uses 3.6 as well. And so there are various reasons for these cutoffs, but we look at 3.6, which also is a little bit more conservative than the other labs. Urinary MMA is an excellent screening tool for low B12, especially in those at risk for neurological disease. This study was specifically looking at people who are suspected to have neurological issues, and then controls. They looked at the B12 status in their blood and the urinary MMA, as well as some other markers. And they found that urinary MMA and serum cobalamin are much more tightly correlated in people who are having neurological symptoms as compared to controls. And so that helps show that the B12 status, there's really something, this is signaling that MMA is on the right track with the B12 status in these neurological patients, but they are less well correlated in the control group. Then that could be because of the cobalamin levels. Those aren't always very predictive, which is why having multiple tests is also sometimes very useful, because it's a pretty complicated process we're talking about. So that's B12. Lots of reasons why it's connected to risk for dementia, and at levels that may or may not be indicative of a true deficiency. So I just was through a few systematic reviews. Their kind of take-home message was that the potential protective effects was strongest with B vitamins. So B12 is included in that, omega-3s and polyphenols. This is looking at a systematic review that looked at all the vitamins and its supplementation trials. And so B vitamins actually did have some of the strongest evidence. This other systematic review found that supplementation of B complex, which included folate, B12, and B6, but particularly folic acid, had positive effects on delaying and preventing risk of cognitive decline. So these are in supplementation settings. There's a positive signal for the B vitamins, and B12 is included, even if it's maybe not the strongest. Here we have a meta-analysis on B vitamin supplementation as well. They found it was associated with the slowing of cognitive decline in people who received early intervention and a long duration intervention. Folate was the most powerful of the three. B12 and B6 were included in this B complex. for reduced risk of dementia in non-dementia aged populations. So here we can see B12, it has, it alone did not have a great effect, but with the other B vitamins, it did. And that to me makes a lot of sense because that's the cycle, the system it's involved in needs all of those all of those nutrients. So B12 is important, but it only is important with other with other nutrients, it seems like. And not only other B vitamins, but I had to throw in a little omega-3 stuff because that's what I know. And this was a study called VitaCog from 2015. And these are looking at brain atrophy rates. So this is a study that they gave high dose B vitamins, including B12, and they measured plasma omega-3 levels, so not the omega-3 index, but at least some kind of omega-3 blood status. So a low blood status is here, these first two columns, and then medium to high omega-3 status are these two. The black bars are the placebo, and the gray bars are the people who received B vitamins. And they did this, this retrospective analysis, and that they found is when they're looking at how much the brain is atrophying, how much the brain is shrinking, there really wasn't a difference between the placebo and the B vitamin group if people had low omega-3 levels. So B vitamins did not help slow atrophy. But if they had medium to high omega-3 levels, then the B vitamins helped reduce the brain shrinkage significantly from placebo. And so this combination of B12 and omega-3 is clearly connected here. The interaction is significant. And it's also thought that the same might be true vice versa, that the effect of omega-3 on brain outcomes may also be dependent on B12 status or B complex status. So this is just one of the hundreds of thousands of nutrient interactions. We need to have good status across the board to really know if we're going to be able to get some of these benefits. And so that's one of the more interesting combination studies. So B12 and dementia, subclinical deficiency is more correlated to neurological issues than the anemias. Homocysteine and urinary MMA are both indicators of low B vitamin status and function. Both do relate to poor neurological health. The B vitamin complexes, supplementation-wise, have the strongest evidence for delaying and slowing decline in intervention studies. But still maintaining good B12 status, measuring the MMA and or homocysteine, showing that that cycle is in good health is an important foundation for long-term brain health and for any of the other potential nutrients. Needing that cycle to be in good health is really critical. Vitamin D. So this is also called calciferol, fat-soluble vitamin. This is mostly, we're getting it from the sun. Fatty fish are the best natural source, but dairy is fortified in the States. Also very, very common supplement. As we know, it's mostly connected to calcium, or we learn about it through basically calcium metabolism. Absorbing calcium, you need vitamin D, and bone health. But as we'll find, vitamin D receptors, so things that if you have a vitamin D metabolite, they would affect that cell. They're found all over the body, and the brain is no exception. So here's a quick look at the metabolism overview. It is fairly complicated, and there are lots of different forms of vitamin D, and some of those different forms are what get across the blood-brain barrier. It involves multiple parts, the liver, the kidneys, affecting bone, lungs, parathyroid gland. This is a body-wide system to get the vitamin D into a functional, into its different functional metabolites. Quickly, clinical recommendations around blood levels for vitamin D. There are two main camps, I would say. The Institute of Medicine focuses mostly on the bone. They look at rickets and osteomalacia as that's the sign of a vitamin D deficiency. That's about 12 nanogram per mil. Less than that is their cutoff. Say 12 to 20 is inadequate. Greater than 20 is basically fine, and you don't need to be above 50. They don't really say an optimal level. The Endocrine Society, obviously its focus is as broader than bone, also focused on chronic disease like cancer and mortality. And from their assessment of the literature, they believe 40 to 60 nanogram per mil is optimal. So they're recommending what's already thought to be excessive by the IOM, 20 to 30 as insufficient and less than 20 as deficient. So less than 12, less than 20 are the two deficiency status ideas. For the OmegaQuant test, we do target 30 to 50 milligrams. It kind of cuts the difference. We are looking at research studies to kind of find the point of the greatest bang for your buck. And it's usually once you're out of the deficiency state, but you don't need to be in a really high range to appreciate some of these results. And to give more context around that, the 30 nanogram per mil, this is a dose response curve. So these are the doses of vitamin D in international units and serum vitamin D in I'm going to talk about this nanomole per mil. For us, if we're recommending 30 on average, that would be about 1,000 IU a day. And that probably sounds pretty low to most people. The IOM also has 4,000 as the upper limit of intake, and we all know that you can take supplements with 5,000 or 10,000, no problem. And maybe that's okay, but for us looking at the research, an amount of supplementation is helpful, maybe necessary for people. But on average, that 1,000 IU, maybe 2,000 should be good for many people. And as we know, there are other things that affect vitamin D levels than just supplementation. So that's where you test, you try something, test, do it for 3 months, and find out what you need to do to hit 30 nanogram per mil. And before we get into the research, I do have to note that there are 2 common units for plasma vitamin D, and it's critically important that you and your doctor or you and your patient know which one you're talking about. In the US, we're usually using a nanogram per mil, but in most of the rest of the world and in research, nanomole per liter is used. They're just different ways of expressing. Nanomole per liter is about 2.5 times higher than nanogram per mil. So this is where I get concerned, is sometimes people will talk to us and they'll say, my doctor wanted me to be at 75 for my vitamin D level. And if we're reporting nanogram per mil, to us, that is still within kind of an okay range, but it's a very high target in our view. And we need to make sure they're not talking about nanomole per mil, because that 75 is equivalent to the 30 that we recommend. And 50 nanomole per mil is equivalent to 20, which is the other kind of adequate status recommendation. Very, very important to make sure you know which unit you're in. So how does vitamin D affect cognition? Like I mentioned, vitamin D receptors are throughout the brain. So these authors suggest that the evidence for vitamin D's involvement in brain function and its ability to affect proteins in the brain known to be directly involved in learning and memory are big reasons why vitamin D affects cognition. So the vitamin D receptor is prevalent throughout the brain, and it might be controlling expression of neurotrophic growth factor and brain-derived neurotrophic factor. Both of these you may have heard of. They are proteins that affect the brain function. NGF is likely to be involved in learning and memory in the hippocampus, and BDNF is affecting survival and differentiation of dopamine cells. And dopamine cells and dopamine signaling is critical in many, many parts of brain health. So those are pretty main reasons why vitamin D could be important here. They also have found metabolites of vitamin D present in the brain and in CSF. So it is crossing the blood-brain barrier. There are receptors for vitamin D. There are metabolites in there. It's doing something. So those are some reasons why vitamin D might help. Let's look at some research. So here we have a systematic review of vitamin D and dementia and Alzheimer's risk. This is let me just see here. Let's see. There were about 2,000 cases of dementia and about 1,600 cases of Alzheimer's disease from 7 prospective studies and 1 retrospective study having over 28,000 samples. So in here, we're looking at hazard ratios for dementia. That line at 1, that's going to be no increase. This is just no risk right here. No increased risk, no decreased risk. So that's where we start. That's our reference point. And then it goes down as we increase in our blood levels of vitamin D. So a 5 vitamin D level is very, very deficient. 10 is still pretty deficient, but you see that more often. We were talking about 20 to 30. So as you can see, the lowest part of the risk for dementia, when you're it's about a 20% reduced risk, is between 20 and 30 nanogram per mil vitamin D. As you get above that, we have a lot less data. There was one study that pulled this back up, and it's not super clear why, but we can see that being in this kind of adequate vitamin D level produced the lowest risk of dementia as compared to the very deficient. Similar to Alzheimer's. There's a little less data for Alzheimer's, but you can see these deficiency. If you're deficient, that's the highest risk level. As you get to 20 to 30, the risk is decreasing by about 20 to 30%. And so that being deficient is that highest risk. Getting to this sufficient level, maybe optimal, that is where we see the best bang for your buck. They also found that increasing vitamin D levels by 10 nanogram per mil was associated with 17% lower risk of dementia and AD. So for every 10 nanograms per mil, you reduced risk by 17%. But you can see these are not linear relationships. That's not going to keep going on forever until you're at 0 risk. It's really leveling out here, it looks like. And they also noted they are not sure what it means when the vitamin D levels are greater than 35 nanogram per mil as it relates to dementia risk. So this is a study using Mendelian randomization, which you may have heard of. It's a new thing in the literature. It's really trying to use these huge data sets we have. So this is using the UK Biobank, which has almost 300,000 people in it. So that is incredible, but it's observational, and observational studies are limited in our ability to assess causation. We can say these two things are related, but we don't know if they're causing each other. They're just related. The Mendelian randomization, through mechanisms that I cannot explain, is trying to get to a place where they can use these huge data sets to determine causal relationships by doing more to control for confounding. So this is kind of a normal observational study. You're going to have your outcome and your variable of interest. You're going to control for some confounders, but in Mendelian randomization, they're doing much more controlling and much more sophisticated statistical work. I'm still a little bit, I want to see intervention studies or some kind of RCT to get to that causation, but it's an interesting development, and we'll see what it's really useful for. But this one is agreeing. This is nanomole per mil, so you can see 50 is 30. This one is agreeing. This is the odds for dementia. One is kind of the no risk here. Increasing odds for dementia if you were below 30, mostly showing up below 10 nanogram per mil. Same story. Extreme deficiency, or just deficiency in the traditional sense, those blood levels are more related to highest risk of dementia as compared to higher levels. But on up to 125, I think that's around the 50 mark. So they aren't even going above 50 in this cohort because it's very uncommon for people to have levels that high. And this cohort, plasma vitamin D was not related to risk for all-cause dementia in a fairly large, over 1,000 people cohort of a Swedish elderly folks. Again, you have that the line at 1, and you can see that the rising vitamin D levels did not reduce risk or really do anything around risk for dementia in this population. So it's not, this is again nanomole per mil. So this 50 mark is the 30. And as you go lower, it's the deficiency. So it's not 100%. A systematic review that was looking at vitamin supplementation in dementia did not, the results for the supplementation trials were not conclusive in assessing the potential benefits that vitamin D might have on cognition. So that's that separation between supplementation and blood levels. They don't always sing the same song. So vitamin D in dementia, the deficiency levels clearly are associated with the increased risk of dementia and Alzheimer's. Sufficient or optimal blood levels have not been determined. They may not exist, but just not being deficient seems to be the biggest hurdle in this story. Supplementation trials, perhaps not effective. And I will say supplementation trials are usually looking in older folks already at risk. You're trying to sort of treat it. If you supplement your entire life, kind of like a low dose 1,000 or 2,000 IU a day, that's not what the supplementation trials are testing. They're testing kind of high dose, shorter time periods, older folks. So that's different. A long-term intake seems to be, for a lot of vitamins, a really good way to prevent long-term issues, and getting that through food, through sun, and if you need to, some supplementation. That's where these long-term trials, we really can find some very striking results. So that's maintaining sufficient vitamin D levels, which can include supplementation, is important for long-term health. Let's go finally to HbA1c and glucose. Quickly, A1c is a hemoglobin protein with a glucose attached. It is, the more glucose is in the blood and in the plasma, the more the A1c hemoglobin has glucose on it. Typically, this is used as a diagnostic for type 1 or type 2 diabetes and to monitor glucose control in people who have diabetes, as not usually tested in people without diabetes. Usually that's just plasma glucose. A1c is super interesting, as you probably all know, is that it gives a good representation of the last 3 months of blood glucose control. And this is based on how long that red blood cell is circulating in the body. But you can see A1c, and I think that's estimated. Average glucose levels are pretty strongly correlated here over the last 3 months. So you can get a good idea of how good their control is with the A1c marker. Plasma glucose will rise and fall throughout the day, as it should. Normative values, HbA1c of less than 5.7 is generally seen as a healthy, kind of normal level. We have a condition of pre-diabetes, which is 5.7 to 6.4%. This is A1Cs starting to creep up. They have a higher risk of developing diabetes, and greater than 6.4% is diagnostic for diabetes. I will note that our test cannot be used for diagnosis of diabetes. It has to be, ours is a screening test, and it would have to be confirmed with another test at the doctor's office. And type 2 is mostly related to this HbA1c. Type 1 also includes other criteria. So high blood glucose, it damages blood vessels, especially blood vessels in kidneys, ears, fingers, and toes. So that's, we hear about amputations, that's that. But anything that's affecting blood vessels, the cardiovascular system, you're also going to have issues with the brain. High blood, it can damage brain tissue over time and cause atrophy in certain regions. This is the dementia piece that we are focused on. Uncontrolled type 1 or type 2 can result in chronic kidney disease, blindness, peripheral neuropathy, and chronically elevated glucose is associated with Alzheimer's, dementias, and CVD. And so this kind of, you know, it makes sense with when we were looking at the picture of the shriveled up brain. If the circulation is not good, if the blood vessels are being damaged, all of this can contribute to the development of these diseases of the brain. So here's a study that was done. This is a meta-analysis, and this has about 20, let's see, it was 122 studies included in this. They found that diabetes confirmed a 1.25 to 1.91 times increased risk for dementia if someone had diabetes. So this is oops, there we go. So just having diabetes is a pretty big risk factor. Prediabetes also showed increased risk, and increased A1C were associated with dementia as well. So they went through and also looked at all the different ways we measure blood glucose, and that was different from study to study. So they used that to also assess which markers were the best at identifying different aspects of dementia. And A1C was fairly good at predicting the dementia state, but also was very good at predicting a mild cognitive impairment that might come right before a full dementia diagnosis. So potentially A1C could be this earlier signal. Certainly that prediabetes state prior to becoming diabetic, that would be a great place to try and intervene. They also found, interestingly, that diabetes medications did reduce risk of dementia. So it's again showing if you're changing this variable, you might get an effect on dementia. And here we have another study where they are taking in many patients. So this is over 20,000 patients in a South Korean hospital, and they're looking at A1C levels over time to measure long-term glycemic exposure and then development of dementia. And what they did here is, you probably can't see it, but these different colored lines, this is called a Kaplan-Meier curve. And every time the line goes down, it means someone has developed dementia. So you want your line to stay up here if you're preventing all the dementia. We know that's not true. So you want the line to be higher up. So this black line, this showed people who had an A1C less than 7. They had the slowest rate of dementia incidence of all the groups, and it is a dose-dependent response. This next line, these are people between 7 and 8 A1C. The green line is 8 and 9. The orange line is 9 and 10, and the blue line is A1C above 10. And you can see that it is a dose-dependent response always helps give confidence that this is a marker that is actually affecting your outcome. It's affecting dementia status, but also the higher it goes, the worse the outcomes. So A1C and dementia, A1C is directly and positively related to dementia risk and AD risk. Long-term A1C is related in a dose-dependent manner. Diabetes to treat medications treating diabetes can reduce the risk. So if you're using medications or lifestyle, either way, reducing the A1C, the blood sugar, is a good way to reduce risk for dementia. And lower levels of A1C, at least less than 7. This is, I mean, we want less than 5.7, but it's again at those extremes are where it really starts to matter. Between 5.7 and 7, it's the more extreme you get, the more higher the risk goes. So often deficiencies, those extreme where we see the real risk happening is often in the deficiency or the disease state. Like for A1C, it's extremely high. For vitamin D, it's extremely low. And for B12, it's actually, that's not as much in the deficiency. So I was mostly thinking about those B12 levels. For all cases, the optimal levels aren't necessarily yet proven. It's mostly what we're seeing is when things are going wrong in the body, that's where the signals are the greatest that there's going to be a disease developing here. And so that's tricky because we want to be able to say, this is your target, and we have some based on a lot of other research. But we also know that the biggest, most important piece is to get people out of deficiency state or out of a very high blood sugar state. And so that is, I think, very true in a lot of parts of nutrition, and it makes, but it does provide, it is difficult, maybe more difficult to motivate on that, but it also maybe allows for a little bit more freedom in what you're focusing on, and not having to necessarily reach an exact vitamin D level, but to know that I'm not in the deficiency state. So that is good. Okay, so that is it for today's talk. We do not have a nutrition talk January 16th. You can contact Kelly and or Tavis. Kelly has a great resource for nutrition questions. Tavis is a great resource if you have questions about using our tests. Again, no webinar in December. Have a happy holiday. And here is the Omega Quant University hub, on-demand nutrition, talks for CEU credit, blogs, Omega Matters, all of it is right there. And this is how you get there. It's our main page, news and resources, right here, Omega Quant University. And that is it. We can do Q&A.
Great, Christina. So if you have any questions, I forgot to mention this at the beginning, but if you have any questions, feel free to drop them in the chat. I'll check the Q&A here. We've got a couple of questions just to start. So first question here is kind of multi-part. I'm curious to know the ideal form and timing of supplementation and how long does it take to restore our B12 levels?
They go up a little bit, but you're talking about B12?
Yep. So she is curious to know the ideal form and timing of B12 supplementation and how long does that take to correct levels?
Yeah, I'm going to have you help me with that one, but I do think it matters why their B12 levels are low. So if they're not eating meat for a very long time, then it can probably restore itself quite quickly. If they have digestion, absorption issues, then you're going to probably be on a bit of a journey to find out what works. Go ahead, Kelly.
Yeah. So as far as form goes, the most popular forms, if we're talking about an oral supplementation, are cyanocobalamin and methylcobalamin. Some people will say there's a slight benefit to the methylcobalamin form. For most people, again, it depends on the reasoning why you're deficient. Methylcobalamin might be slightly more effective than the regular cobalamin form. For patients who have like those digestion issues, they are going to be requiring B12 injections. So that would be that. And then the second part of her, as far as how long it takes to restore, yeah, that can be very variable. It really depends on the person. If there's not a digestive issue and someone just needs more B12 in their diet, it shouldn't take too long to restore levels. I would guess somewhere in the 4 to 6 week time frame. That's why testing is important, because that's a very individual thing. Which is the best test to request from a healthcare provider in regards to B12, either blood or urine? I think she's talking about MMA.
Yeah, there are a lot of ways you can test for B12 status, and like I said, you're really wanting to look at the health of that cycle. So RBC folate is something people test. Serum cobalamin is the most common, I would say. But you can get MMA either in plasma or in urine. And plasma MMA can be more affected by kidney function. So that's where the urine one isn't affected by kidney function. A combination, like this is still an area of, I would say, debate as to what we should be testing. The newer research is really, there's a lot of interest in the MMA as being a very specific marker of B12, because homocysteine can also be affected by a lot of other things. And serum cobalamin is by far the most common. So that's going to be the one the healthcare provider most likely knows to test. The problem is it doesn't always match with the symptoms. And so that's where the other argument for MMA is that it is indicating the function of B12, not just the level of B12. And so because it's a signal of that cycle. So homocysteine MMA give that information. Having all of it is also great, but it is a little bit, I'm not an expert in this, so I'm sure there are other opinions on it, but I would say serum hemoglobin is the most typical. You get more information other places.
Yeah. What is the relationship between low vitamin D and cancer risk?
So that would be a different talk. Typically, we see very similar patterns with vitamin D levels over time and risk for cancer and other chronic diseases. Having non-deficient vitamin D levels, a little bit higher vitamin D levels over time usually corresponds to a lower risk of cancer. But I do not have the studies in front of me to confirm that. It's just that's a pretty fairly good consensus, but it's, again, that's cancer and Alzheimer's diseases are ones that are tricky to talk about because they are both ones that feel like there's very little you can do. You're just going to get it or you're not, and they're really scary. And it's not as if, oh, I take this one supplement and I'm protected for the rest of my life. It's all a part of the entire process, and vitamin D status is part of it. But sometimes people want to take more. More is better. More is better. What I want to say is more is not always better, but having enough is very good. So that's my little soapbox.
Is there any recent evidence about the association following a strict vegan diet with dementia? I think this would probably be more in relationship with like the B12 and things.
Those papers were popping up and I didn't look into them, so I don't want to comment. My one thing would be like, there probably hasn't been enough people on vegan diets for their whole lives who are old enough to have dementia to have a lot of data on it. But do you know, Kelly?
Yeah, I don't know of any either. I mean, obviously with a strict vegan diet, you're going to have holes as far as some nutrients, specifically B12 is going to be your biggest one. So that would have to be addressed in order for it to be a balanced diet. As far as like the other nutritional components, I would like to point out that you can eat a strict vegan diet by eating nothing but Skittles all the time. Those are vegan. So just because a vegan diet, and I say that obviously to be, sort of a little inflammatory, just because a vegan diet doesn't always mean it's healthy in terms of like nutrition and blood sugar and all of that kind of stuff. So, and there can be big holes. So there's probably a lot out on that.
Yes, but vegan diets, vegans and vegetarians do tend to have better cardiovascular disease metrics, and that's very important. And if they're supplementing correctly, it's could very much be a very good diet for that. If that's what you want to do. So it's also, I'd be interested in the studies coming out is how much do they know about the supplementation in these people's diets. So good question. Maybe we should do a talk on vegan diets because so many of the metrics we look at are animal based, and that's not intentional. It has just happened that way. And it's interesting. So yeah, it's a good question.
So there's two more questions here that kind of relate to each other. So doesn't genetics influence utilization of B12 and folate, like the MTHFR gene?
Yes. Yes, absolutely. There's a huge amount that we can't necessarily control and how our body uses these nutrients. So that's where I'm not deep into this area like MTHR and all that MTFR. So I don't have a great answer on that, but in all of this, genetics can play a role. In all of this, there are people with certain genetic characteristics that give them this extreme profile in their blood levels and their symptoms that show us what those genes are doing. But those, how typical those different types of genetic polymorphism polymorphisms appear in our population in the US and populations across the world. Those are, there's definite patterns across the world in how people process things. And it's now that we've all moved around, it is messy. It's very messy to do research in this area. But that's part of we can't change that part. We just kind of have to deal with it. And I don't know what that would do to certain blood levels. If it's like if you have high blood levels, but you're not actually using it because of a genetic polymorphism. There's definitely genetic tests around this. So maybe you start to combine those kinds of information. But
yeah,
that's getting pretty deep.
Yeah, there's a lot of conflicting information out there as well too about the MTHFR gene and how it processes folate or affects folate processing and all that too. So there's a lot. That's a whole nother can of worms.
That's a whole nother talk for sure.
Yeah. We're making notes. Every time we do one of these, we make notes on future topics. So these are great questions. All right. Do we have any more questions? Anything else?
B12 levels would be higher. That makes sense.
Yep.
And then 25% of Hispanic population. Yeah, that's interesting. And it's also the Hispanic population, they will have certain genetic polymorphisms that track with them. So this is great information. Thank you guys for putting this in the chat. And this is how personalization of medicine and nutrition. We're learning so much more, but how do we utilize that every day when we can't have every piece of information that we might need? So it's important to definitely learn more about this and also keep balanced with some basics of nutrition that work for most people. It's good stuff.
Yeah. All right. One last call. Are there any more questions at all for Christina before we wrap it up today? All right. That looks like all we have. Thank you so much to everybody for joining. Again, just a reminder, there will be no nutrition talks in December. Everyone rest up and relax for the holidays. And but we will see you again in January, January 16th at same time, 1 p.m. Eastern time. In the meantime, go ahead and check out Omega Quant University. And again, check out our Black Friday and Cyber Monday sale between the 20th and 27th of November for that 20% discount on our tests. So thank you so much, everyone, and have a great rest of your day.